first year in university implementing sort of filtered flexibility. You have that SAP, that student absence portal that you can do documented and non-documented absences. And because of a number of reasons, there's not enough prompters to do in-person tests. So because of that, things have to move online. So putting everything online in this big test is sort of what we're dealing with at the moment, and all our tests are online, and we're trying to figure out what's going on with that. So with test one, our first test that we had, average was 89%, right, which is high. Most people will agree that's a little bit high. I don't think people's expectations are. Hold on, there's someone's getting a phone call here. This will help you guys remember to turn your phones off next time. 89% is unreasonable. I think you guys agree with that. It was too high. It was the first test we're trying to deal with this online, no proctoring sort of stuff. When we looked into it, that test was sort of vulnerable to AI. So if you put in the questions, AI spit out the answers pretty well. So going into test two, we appreciated that things are going to be different. So Dr. Cumming kind of wrote the test using more personal stuff that we say in lecture. So if it's said in lecture, AI doesn't necessarily know about it, and therefore it can be done a little bit better. However, for that test, there was rampant cheating going on, so lots of people got perfect, so we eliminated that test. However, if you look at the distribution, I showed you a graph of that, where there's 584 perfects, but then otherwise there's a normal distribution. If you sort of look at just the normal distribution, the average on that test would have been about 74. The average on the test that we just wrote was about 75. It too was pretty AI-proof. If you used AI on that test, you would have scored probably 10 or 11 out of 20. So, don't use AI. Hold on here. Other than dropping it on the floor, how do you turn off an apple? Stop. All right, there we go. It's okay. Maybe just throw it away.
There's like 5 timers on. Jeez. There's like 50 on.
All right. So, this recent test has an average of 75, which is in line with the averages that we've had over the past many years in this course. Averages are usually between 73 and 77 sort of stuff, sometimes in the low 70s, sometimes in the high 70s. Things tend to work out. I think students sort of didn't like the test because the average is much higher than the first test, obviously. A reminder, your top 3 out of your 4 count. All right, so not everyone should be expecting 100% on their test, and be reasonable with things. I think you guys have to learn how to learn. I think that's one of the issues. I've had lots of emails saying, I'm really struggling with this. How do I do things? And I think you have to appreciate that all of these questions are basically true and false questions, right? Multiple choice is nothing but true and false. So even if it's multiple, it's true and false. Is the fact true or false? There is no room, well, maybe this way, or if I consider it like this. No, it's true or false, right? So when you're looking at these questions, each answer, true, false, true, false, true, false. There's no if or that, right? And given that, only one answer is correct. That means 3 of them or 4 of them or whatever are false. So pick out the crap. I think you guys are wary of doing things. You have to be good at recognizing the crap. We write a question, we write an answer, and then we have to fill it out with something else, something that's going to be a distractor, something that has to be reasonable, but it's up to you guys to recognize what is false, right? The best way to do that is to organize the information into categories, or into groupings, or into processes, or into functions. There's many ways to put together the data that I'm giving you. I think on the last test, we went through 42 PowerPoint slides. Right, so all that material was on 42 PowerPoint slides. It's not a lot of information. There's hardly any words, hardly any topics in there. It's an open book test. Organize things, have a piece of paper, write things down. As we get into Actin, it's going to be even easier. Make little categories, motor proteins, maps, signals, sensor zones. What's in a sensor zone? So you know what's in a sensor zone. You know what's in an actin. This protein is associated with this function and this only. Right? An actin is only going to be found in an actin. Okay? Put things into categories for yourself so that you have these things, and so when you get to the question, you can recognize all the garbage in the question. I think that's where people are struggling. You're kind of looking at some of the answers saying, well, maybe this is possible. There's no maybe, right? If there's a maybe, it's because you didn't study or you're misunderstanding something. There's one answer, and the rest of it is just filler. Recognize the filler. It's really easy. Okay, you guys are probably frustrated with your grades on the test that we just had. There's 20 questions. I think 7 of the 20 questions, more than 90% of the students got them right. There's only 6 problematic questions on the test. Problematic in terms of students sort of didn't get perfect on them, so to speak, right? When you look at the questions and the statistics, though, the statistics tell us the story. If we write a bad question, the statistics will scream out it's a bad question. There's 1,200 of you writing the test. If there's a question that's bad, it'll be very obvious that it's bad. Right? So we get everything back. Every little click that you do, when you logged on, when you started a question, when you submitted a question, right? Every single time point is given to me, and then you get all kinds of other data back, such as this. So there's lots of things. So in this example here, you can see how many people picked the correct answer, and there's a whole bunch of other stats that come with this, in addition to other answers. So there's a measure called the point by serial. The point by serial tells you how the students did on other questions compared to this question. Right, so it kind of gives you whether this question is relevant or not, and kind of leads to this discrimination index. So this tells you if the student did badly on other questions, how did they do on this question? And that goes from -1 to +1. So -1 means there's something wrong with this question. The student did excellent on these other questions, and suddenly they're getting this wrong, and other students are sort of based on other students as well. So things like this tell you how good a discriminator of the question is. If it's negative, so it goes from -1 to +1. If it's negative, it's a bad question. There were no negative questions. All the point by serials were fine. They were strong. Anything sort of above 0.2 is good. So I know the questions are good. I know students get frustrated with sort of looking at stuff and going, but that question was hard. I don't know what to say other than saying that the stats scream out at you if there's something wrong with the question. The weakest questions in terms of point by serial are questions that were too easy. Right? So you get questions like this, where almost everyone gets the question correct, and it has a point by serial, and it's not a good discriminator. It's not a good discriminator because it's too easy. Students that did badly on other questions are actually getting this one right. So there's tons of stats out there that tell me that the questions are okay in terms of the way people are answering them. You just have to figure out how you are looking at things and why you're choosing what you're choosing. Things like CDK phosphorylation. When the heck did I mention CDK? CDK was mentioned once, and that was in the context of maps. Right? So if you put things in the category CDK, map phosphorylation, has nothing to do with thymine and kinesin. So look at questions and answers and pick out the ones that say, no, it has nothing to do with anything. Minus and microtubule occurring in both sensor zones. The number of times I've said these things are not called sensor zones at this point. These are not sensor zones. These are the poles. Right? So I know there's only a few students that are answering these particular questions, but my point is, usually the answers scream out which is right and wrong. Yes, some of them are more difficult than others, but in general, don't be disappointed in the way you did. An average of 75 is great. I know everybody wants higher grades. Hopefully you get higher grades. In principle, everyone should get 15 out of 15 in the tutorials. You're doing 3 out of 4. on these tests. Your grades should be hopefully okay. The final exam, which I'll talk about in a second, will be tougher, but we'll get to that when we get to it. All right, this past test. Students who wrote in the last third of the 10-hour window tended to do better. All the students who had 100% on this last test finished in the last 3 hours. Also, all the students who did well and wrote in the last few hours tended to write the test quickly. Okay? What's that proof of? I don't know. Certainly, I can look at all the stats and I can see all the students that are doing this, and I'm keeping a track of the students who did this first test and second test and stuff like that. Is that truly anything? Probably not. In principle, the simplest solution to fix this is to shorten that window. Right? It looks like students are just chatting with each other. I mean, I have screen captures of people sharing screen captures of questions and trying to figure things out. Why you would do that, I don't know. Why you're helping others to do better than you. Regardless. So in principle, a shorter time window is sort of a way to solve this. In reality, it's too difficult to implement. So it's going to stay the way it was. So the next two tests, the time window is from 12 noon to 10 p.m. on Friday. All right? So there's job problems, Ramadan problems, math test problems, other problems that are difficult to deal with. However, if you guys are aware of people doing nefarious things in that 10-hour window, please share them with me. Okay? Is that all right? That's a long way to say all that. Tutorial videos 3 and 4, I'm sorry, I don't know what to say other than I'm sorry, it's embarrassing, I'm on it. I still have emails to go through, so lots of people emailed me about jobs or Ramadan or math tests or other issues with that time window. Hopefully that's dealt with because that time window is gone now. All right. So, that's it for that. Let's get back to our learning. So we're going to have the last 3 slides on microtubules, and then we're going to go into actin. All right, so this was the third last slide in section 5, I think, or 6. And we were talking about the mitotic apparatus and sort of how there's some things going on in the mitotic apparatus to move things around. So you capture all the chromosomes are connected to the kinetochore from both poles. And now you're trying to center this mitotic apparatus, you're trying to center it into the cell. And you're using these things called cortical anchors. And what's in those cortical anchors is dynactin. If you remember, dynactin is what dynein, the motor protein, uses to bind to cargo. And in this case, it's going to be binding to the astromicrotubules. Right, so the astromicrotubules and dynein and dynactin are bound together at the plasma membrane on both sides. So basically, dynein is bound to the plasma membrane behind the poles. There isn't any dynein in the middle. There isn't any in the metaphase plate. Right, so that dynein can act on the astromicrotubules on one side or on the other side. So it's basically pulling the astromicrotubules towards the right or towards the left, one side or the other. Right, dynein is a minus end-directed motor protein. The minus end is at the poles. So if you activate this cortical anchor on the right side, the dynein on the right side will pull this astromicrotubule, and it'll pull it towards the plasma membrane, and it's pulling this whole mitotic apparatus towards the right to center it. Okay, as you can see, because this astromicrotubule is close to the plasma membrane, as it's being pulled, it has to be depolymerized, otherwise it's going to poke through the plasma membrane. There's a whole bunch of other proteins involved here, involved in activating and inactivating things. You don't have to worry about them at the moment. But what is important, though, is that there is nothing present in the metaphase plate. Right? There isn't any dynein in the metaphase plate. Okay, so dynein is used to center this mitotic apparatus. So once it's centered, that's metaphase. After metaphase, you have anaphase. And I started going through this, and I'll go through it again this time. Okay, so anaphase is separating the sister chromosomes so that each sister chromatid goes to the opposite poles. So there's a lot of things that have to happen here. Cohesions have to break, and there's a whole bunch of other mechanisms that we're not going to deal with. What we're dealing with is the microtubules pulling the sister chromatids apart during karyokinesis, which is separating the DNA. So, and there's two phases, anaphase A and anaphase B. Anaphase A is the movement of the chromosome toward the pole, so the distance between the chromosome and the pole shrinks. And anaphase B is that the distance between the poles themselves is bigger. So the poles are being pushed apart as the chromosomes are moving towards each of the poles. It's funny that it went on. Okay, when you look at anaphase A is driven by microtubule depolymerization. So on this little diagram here, it has an A1 and an A2. So that is when you look at the kinetochore and when you look at the pole, you can see there's depolymerization of that kinetochore microtubule at both sides. So both at the plus end and at the minus end of that kinetochore microtubule, there's shrinkage. There's depolymerization at both ends of that kinetochore microtubule. That kinetochore microtubule gets shorter. As that kinetochore microtubule gets shorter, that chromosome moves closer to the pole. So two things here. One is, well, if that kinetochore microtubule is getting shorter, how is that chromosome still attached to it? It is. All right, you got to remember, I'm going to pull up a picture from before, that the plus end of the microtubule is free. The kinetochore, there's an outer and inner kinetochore. The plus end of the microtubule is free, not like it's drawn here, right? So if you're depolymerizing this plus end and you're shrinking this kinetochore microtubule, these proteins in the kinetochore can actually still bind to the microtubule and move along toward the pole, right? So the kinetochore proteins are still attaching that chromosome toward that kinetochore microtubule and moving it toward the pole. This is a redundant mechanism. There are motor proteins present here. Motor proteins can also do this. The important part, though, is if you inhibit motor proteins. So if you inhibit motor proteins, anaphase A still continues. So the chromosome will still move towards the pole in anaphase A, even if you inhibit motor proteins, because depolymerization alone can drive that. Okay, so it's just the depolymerization that's not just, but depolymerization on its own can drive kinetochore microtubule shrinkage and movement of the chromosome to the pole. Another thing to note here is that at the minus end here, by this, the pole, the spindle pole, which is the microtubule organizing center, there's also depolymerization at the minus end at the spindle pole. This is different than in a centrosome. You should remember in a centrosome you have a gamma tubulin at the minus end that doesn't allow minus end depolymerization. That's why this is not a centrosome, it has different properties. The spindle poles allow minus end depolymerization and it occurs during anaphase A. So that's another sort of reason why this is not a centrosome per se. It's a spindle pole, has different characteristics than a centrosome. The most important being, or one of the most important being, that you have minus end depolymerization, which is crucial for the function of the kinetochore microtubule. It's causing that kinetochore microtubule to shrink. Okay, so you have shrinkage of the kinetochore microtubule that's pulling the chromosome toward the pole in anaphase A. Make sense? And that doesn't require motor proteins. If you inhibit motor proteins, that still occurs. That's in contrast to anaphase B, and that's the movement of the poles apart. So as the chromosome is moving toward the pole, the two poles are separating. And they're separating because of two reasons. One is because of polar microtubule sliding, and the other one is because the astral microtubules are being pulled by dynein. Let's talk about the astral microtubules and dynein first, because we just talked about them. If you remember, you have dynein present as part of these anchors that are involved in centering the mitotic apparatus in the first place. So you have dynein anchored on either side. It's already present. It's involved in centering things. During anaphase B, you can trigger that dynein again, except now you're triggering it on both sides. So the dynein that's sitting on the plasma membrane is going to try to walk to the minus end. As it's trying to walk to the minus end, it's going to pull that astral microtubule towards the membrane. So the astral microtubule and the pole is moving, in this case, to the right, and on the other side, it's moving to the left. So the poles are moving apart because astral microtubules are being pulled by dynein, pulling the poles apart. At the same time that the poles are being pulled apart, they're actually being pushed apart by kinesin 5. So as part of the mitotic apparatus, you have these things called polar microtubules. Polar microtubules are microtubules that came from the opposite poles. They didn't find a kinetophore, but they overlap with each other. Right? So there are overlapping polar microtubules present in the mitotic apparatus. In between those polar microtubules is kinesin 5. If you remember, kinesin 5 is made up of 4 heavy chains. They don't move cargo, but they have 2 head domains on either side. And when you put them in between these polar microtubules, the kinesin on one side is going to move to the plus end, kinesin on the other side is going to try to move to the plus end. So the relative motion is that you undo that overlap. So if you can imagine, that kinesin is not going to move because it's just pulling things in both directions. But what happens is that overlap that was present between the polar microtubules will get undone. So if you can think of these polar microtubules as stiff rods, as you're undoing those polar microtubules, you're pushing the poles apart. So the kinesin 5 is pushing the poles apart, while the dynein on either side is pulling the poles to the opposite direction. So that's going to cause this big empty space where the metaphase plate was. Make sense? And that's because in this place where the metaphase plate was is where you're going to undergo cytokinesis. Actin is going to form a ring here, and this is where you're going to start pinching off the cell. And where that pinching occurs is you want to make sure that none of these sister chromatids are anywhere near that, because you don't want sort of inappropriate segregation of these chromosomes. You want the chromosomes to be very far away from this metaphase plate when cytokinesis is occurring. And to do that, you're both pulling the chromosomes with anaphase A and pushing the poles with anaphase B to get everything away from the center. Okay? So, anaphase A and B, anaphase is finishing up. You're going to start forming new nuclei in telophase, and eventually you're going to start undergoing cytokinesis. Cytokinesis involves the formation of an actin ring, which we'll start talking about actin in the next slide. And there's a lot of stuff in here. You don't need to memorize most of this. What's important to appreciate is that that contractile ring, that actin ring that's going to be responsible for cytokinesis, is going to be present where it is due to the presence of this Rho protein, right? So the Rho protein is localized where the polar microtubules overlap. So when you form that mitotic apparatus and you put that mitotic apparatus in the middle of the metaphase plate, the presence of those polar microtubules resulted in sort of this localization of this protein called Rho. And that Rho protein is going to be involved for localizing actin and localizing the contractile ring in where the metaphase plate was. So the polar microtubules are the center of the metaphase plate. They're going to localize a protein called Rho. Rho will then localize actin and contractile ring, and that's where cytokinesis will occur. Cool? So that's the end of microtubules, and now the beginning of actin. So I don't know if you guys need to shift things. Okay, so all cells have actin, tubulin, and intermediate filaments, so they often work together, and we will see how they work together in several different examples. Actin, as I mentioned before, comes first in the textbook, so a lot of the stuff we talk about with microtubules, we're going to revisit with actin, things such as nucleation and critical concentration, except now we're going to go into more depth because there's more material on actin. Right? Actin is cortical. What does that mean? That means it's towards the outside, the cortex. So the cortex of something is the outside region of it. Actin is often associated with the plasma membrane. Okay, because it's cortical, because it's associated with the plasma membrane, it can cause changes in shape and movement, and we'll talk about changes in shape and movement. And you can see different cells here moving around different ways. So actin is involved in movement of a variety of different things. Okay, so it's at the cortex, at the plasma membrane. It can influence this plasma membrane to cause movement, but because it's also at the plasma membrane, it can be involved in many cell signaling functions. Right, it's at the plasma membrane where you have lots of receptors. So actin in a lot of processes can be mediated or can mediate the actin cytoskeleton both ways. Actin can mediate things, but other things can also mediate actin. Right, we're going to talk about lots of different structures with actin. So this is sort of an overview slide of things that we'll talk about with different things that actin can do. Again, most of these are cortical. Most of these are associated with the plasma membrane. So microtubules are sort of deep inside the cytoplasm. They come from a deep centrosome and they move towards the outside. And then actin is found at the plasma membrane. So in general, microtubules move things to the plasma membrane, hand it off to actin, and then actin can move things along the plasma membrane as needed. Actin can form different structures, many more structures than tubulin can. It can form both networks and bundles, and we'll talk about what they are in a second. So networks are basically parallel aligned with things, versus networks are cross-linked things. So we will talk about microvilli and how actin is found in the cell cortex and these structures called adherens belts. We'll talk about migrating cells and structures such as filopodia, lamellipodia, and stress fibers. And we'll talk about muscle function, but also non-muscle function, things such as phagocytosis, endocytosis, and of course the contractile ring during cytokinesis, which we just alluded to. So actin is very dynamic. Microtubules are dynamic, actin is even more dynamic. So it can form bundles and networks, and if you look at a cell, so this is a cell with fluorescently labeled actin. You can see that it has bundles, and this sort of faint glow here is a network. So as you see this cell will move in a second, you can see it's going to form new bundles and it's going to form new networks. Right, so it's constantly very dynamic with bundles and networks being formed. The networks are the fuzzy sort of area, and the bundles are those more pronounced things. When you look at images of any migrating cells, you can see all kinds of striations that form because of actin dynamics. So actin is forming networks and bundles as it moves along, or as it's polymerizing and depolymerizing. All right, bit of an overview now, stuff to memorize. Okay, actin is made up of a monomer called G-actin or globular actin. Okay, so G-actin is the monomer. It doesn't form a dimer, it's just a monomer that polymerizes. Similar in naming, there's an alpha, beta, and a gamma actin. Just like there's alpha, beta, gamma tubulin, there's alpha, beta, gamma actin. These different types of actin basically form the same microfilaments. So we're not really going to distinguish between alpha, beta, and gamma actin. You can see up here that alpha tends to be found in muscle, beta is found in the cortex, gamma tends to be found in stress fibers, which are associated with cell migration. But they can be found, multiple actins can be found in one cell type. So there's nothing unusual about a cell having beta actin in its cortex, and also gamma actin as it's migrating. So we're going to ignore to some extent that there are different types of actin. Actin is actin, it's a monomer. Okay? It's a monomer that has 4 domains, so you can see, 1, 2, 3, 4. It's almost like a clover leaf, has 4 domains. However, it's not symmetric in that on one side of it, it has an ATP binding cleft. So it's this 4-lobed protein that one side has a cleft in it. So that cleft gives it polarity. So the monomer has polarity. When the monomer polymerizes, all of the monomers line up in the same orientation. So when you polymerize a microfilament, that microfilament has polarity. Okay, so the G actin, globular actin, polymerizes into F actin, which is a microfilament, and that microfilament has polarity. So these little red balls here are meant to represent the G actin. And you can see this little slit present in all of these. That slit represents the ATP binding cleft. So that's just showing you that all of these actins are in the same orientation. So they're all in the same orientation, and because of that, actin has a plus end and a minus end, just like tubulin, and you'll see that's associated with polymerization. Okay, so G actin polymerizes with the F actin. All the monomers have the same orientation, polarity, and you form this microfilament, F actin. It does so, though, in a double helix. So you can see from this structure, these actin monomers come together to form a double helix. So the F actin is a double helix. It has a certain periodicity, so you can look at how often this double helix bends around. And so these little asterisks are trying to show you the monomer sort of going around, and a monomer to be present from sort of on the double helix facing you to be facing you again is about 72 nanometers. Right? So a complete turn of that double helix is about 72 nanometers, where 36 nanometers is where one monomer is facing you that's from one of the alpha helical chains, and at 36 nanometers it's the other monomer that's facing you. So 36 is half a turn, 72 is a full turn of the double helix. And again, that's important when you're looking at step size of motor proteins, trying to understand where things are going. So actin has polarity, it has a plus end and a minus end, and we've known this for many dozens of years, maybe even over 100 years, because you can do these manipulations with something called myosin S1. So myosin is the motor protein, and you can degrade the motor protein to form something called S1. And we'll talk about that when we talk about myosin later on. So you can use part of myosin, and you can add it to F actin. You can add it to filamentous actin. So when you add this fragment of myosin called S1 myosin to filamentous actin, it forms this pattern. And people call this pattern an arrowhead. So this is looking under microscopy. There's this pattern where it looks like there's little arrowheads pointing, and those arrowheads are always pointing to the minus end. So this is called decorating F actin with S1 myosin. So when you decorate actin with S1 myosin, you get these little arrowheads. You can always figure out where the minus end is because the arrowheads are pointing to the minus end. The barbed end, which is the open end of the arrow, is at the plus end. Okay, so you can use S1 myosin to always find the minus end of actin. Interestingly, when you coat filamentous actin with S1 myosin, you also stabilize that little fragment of actin. So now this little piece of filamentous actin will not depolymerize because it's coated. So it can't depolymerize, but it can still polymerize. So you can use this coated fragment, this S1 coated fragment of filamentous actin as a nucleus, because you have this thing that won't depolymerize, but you can have it to polymerize. So you can do experiments such as we saw with microtubules, right? You can put it in an environment and you can start to add actin monomers to it, and after a certain critical concentration, you'll get growth. You'll get growth from both the plus end and the minus end. So above critical concentration, the plus end grows faster, that's why it's called the plus end. Below critical concentration, you won't get polymerization. The plus end grows faster than the minus end. The plus end depolymerizes faster than the minus end. So if you're above critical concentration, you grow. If below critical concentration, you depolymerize. Make sense? Same thing we talked about before, and we even used this slide, the concept of a nucleus. If you have a nucleus, you're going to get the elongation faster. If you don't have a nucleus, you have to form a nucleus from the G-actin or other sort of subunits. And we'll talk about other nuclei in a second. Okay, so same concepts as before. Above critical concentration, you will polymerize. However, if you don't have a nucleus, you have to make a nucleus first, so therefore there will be this nucleation time, this lag phase before you get to this quick growth. Okay, so above critical concentration, you polymerize. Below critical concentration, you depolymerize. However, a bit of a twist in that the critical concentrations of the minus and the plus ends are not the same. When you look at the critical concentrations, you can see that the critical concentration of the plus end is 0.12 micromolar versus the critical concentration of minus n is 0.6 micromolar. Okay, so the critical concentration for the plus end is lower than the critical concentration for the minus n. Right, 0.12 is about 5 times less than 0.6. So to kind of recap, if you're above both critical concentrations, you will polymerize. So if you have 0.8 micromolar subunit, so you have G-actin at 0.8 micromolar, 0.8 is bigger than 0.12, 0.8 is bigger than 0.6, you will polymerize on both. If you're below the critical concentration on both ends, if you're 0.05 micromolar G-actin, you will depolymerize both. Right? And there are numbers up here you don't have to memorize, that polymerization and depolymerization is faster on the plus side, slower on the minus side. For microtubules, it's the same thing. The critical concentrations for the plus and minus ends are not exactly the same, but for microtubules, it almost never matters because it's almost always just the plus end that's doing things. The minus end is just stuck in the centrosome down to gametubulin, or it's in a basal body that's been sequestered in the bottom of a ciliary flagella, right? So in most times, the minus end is not important for microtubules. Having said that, we just talked about, anaphase A, but that's a unique situation. But for actin, it's not capped at either end. It does have an open minus and plus end, so it's important that it has a different critical concentration at the minus and plus end. And that's because if you're at a concentration in between the two critical concentrations, you can get something called treadmilling. Right? You have a plus n critical concentration of 0.12, a minus n of 0.6 micromolar G-actin. What happens if the G-actin concentration is 0.4 micromolar? So 0.4 is bigger than 0.12. So you can add to the plus n. So the plus n will grow because you're above critical concentration. However, 0.4 is less than 0.6, so you're going to be depolymerizing to the minus n. So if you're in between the two critical concentrations, you're putting monomers on one side while you're removing monomers from the other side. And when you look at this filament, it looks like this filament is treadmilling. It looks like it's moving to the right in this case because you're adding on one side and removing from the other side. So in this example, they kind of give you these blue labeled monomers. So these are just to kind of orient you to this position. So you have this little microfilament here. You add a critical concentration between the two critical concentrations, so let's say 0.4. So you're adding to the plus n. So the distance from these blue monomers to the plus n is growing. right? So it looks like these blue monomers are moving away from the plus end. At the same time, you're removing monomers at the minus end because 0.4 is less than 0.6. So the distance between the blue monomers and the minus end is shrinking. So it looks like it's growing on one side, shrinking on the other. It looks like this microfilament is treadmilling. It looks like it's moving to the right, and indeed that's sort of what's happening. And this is an important phenomenon in cells where it's adding to one side and growing to the other side. Okay? For those of you who are astute, you'll notice that you're adding on one side slower than you're removing because the amount that you can add on the plus side is less than you can remove on the minus side. So the amount that you're growing is not the same rate as it's shrinking, but in principle, you're doing it sort of not at the same speed, but at least you're growing at one end while you're shrinking at the other end. How do they navigate without a tether? They move different things. We'll talk about how they move things when we get to them. So that'll be a confusing thing when we talk about endocytosis and vesicular transport. Okay, so far so good? All right, so there's different critical concentrations, and memorize those. 0.12, 0.6. Memorize them. When you look at a cell, and look at the amount of G-actin in a cell, isolate G-actin from a cell and do calculations, you see that a cell has about 400 micromolar of G-actin in it. I just told you the critical concentrations were 0.12 and 0.6. There's 400 micromolar inside the cell. Why isn't all of this polymerized? It's because it's all basically kept as a reservoir to allow rapid polymerization when needed in different locations. So there are three proteins, there's three proteins that are central to this that you guys need to memorize. Okay, there's a protein called thymosin, profilin, and cofilin. Thymosin is very abundant, and it basically binds to G-actin and takes it out of that critical concentration formula because it does not allow it to polymerize. So most of the actin in the cell is bound up by this protein called thymosin, and thymosin sequesters this actin. So there's a thymosin cycle basically where thymosin will bind to most of the G-actin and then release some of it as needed. We're not going to go into those mechanisms. But most of the actin is bound to a protein called thymosin. Another important factor that we didn't really talk about, we'll talk about now, I guess, is that the G-actin that has to be involved in polymerization has to be in the ATP form. So we mentioned actin has an ATP binding cleft. It needs to be in the ATP form to polymerize. If it's in the ADP form, it cannot polymerize. So it doesn't matter what the critical concentration of the G-actin is if it's not in the ATP form. So profilin is a protein that's involved into energizing that actin. So if you don't have profilin acting on actin, then it doesn't matter what the critical concentration is, it just won't polymerize. So just like with the microtubules, the ATP is hydrolyzed after polymerization. We're not really going to talk about that, but ATP is hydrolyzed after polymerization. Okay, so thymosin is sequestering. Profilin is providing the right energy state, the ATP form for the G-actin. So that's kind of keeping things from polymerizing or allowing polymerization to occur. Another protein is a protein called cofilin, and cofilin acts to depolymerize. So if you have a lot of cofilin, it's going to fight all this polymerization by basically causing depolymerization of the actin microfilaments. All right, so if you're above critical concentration, thymosin has to release you, profilin has to activate you in the ATP form, and then ideally there shouldn't be a lot of cofilin around. Once you're depolymerize, the depolymerized variety is in the ADP form, and it has to be energized to get into the ATP form. So again, just like with microtubules, this ATP caps that sort of present that's eventually energized down into ADP, hydrolyzed into ADP. Okay, so thymosin, prophylin, cofillin. We're not really dealing with exactly how they work in terms of how thymosin is regulated or anything. Okay, so another way that the cell can regulate polymerization and depolymerization, has all these monomers, is by capping the different ends of the growing microfilament. So if you have a microfilament, there are proteins present. One is called CapZ, and the other one is called propomodulin. So CapZ can cap the plus end. So in the presence of CapZ, the plus end is going to be capped, and you will not get any polymerization at that end. Doesn't matter what the critical concentration is. If the plus end is capped, that only leaves the minus end to function, to polymerize or depolymerize, and the minus end, if you remember, has a critical concentration of 0.16. Oh, sorry, 0.6 micromolar. So if you have something capped with kappa Z, it's the minus end that can grow at a certain critical concentration. There's another protein called propomodulin. Propomodulin can cap the minus end. If you cap the minus end, then the plus end can polymerize and depolymerize. It has its own critical concentration, that's 0.12. So you can regulate what the growing microfilament is doing using CapZ or propomodulin. If you cap both, then you just have a stable microfilament that isn't going to grow on either side or depolymerize, won't polymerize or depolymerize. Okay, so those are natural proteins found in cells that are used to look at, used to stabilize used to stabilize actin. There are also actin drugs that we use in studies. Right, there's a drug called cytocolazin that depolymerizes actin filaments. So if you add cytocolazin to a cell, you will depolymerize the actin cytoskeleton. There's another drug called colloidin. Colloidin binds to and stabilizes the actin filaments. Again, just like when we talked about with Taxol, it can do things at different doses. For our purposes here, it's not going to depolymerize. Okay, colloidin, when you add it to a cell, it will not depolymerize the actin. You may recognize phalloidin because you can also make phalloidin fluorescent. Phalloidin is a drug that binds to actin and stabilizes it, keeps it there, whatever size it is. If you make it fluorescent, you can then add it to a cell and then look at the actin network, which people do all the time. All right, so we have CapZ and propomodulin that affect stability, as do drugs, cytocolazin and phalloidin. Okay, so you have lots of actin monomer in the cell. You have 400 micromolar, way above the critical concentration. So it is wanting to polymerize. So you want to regulate where polymerization is going to occur. That's sort of what the centrosome does for microtubules. The centrosome is basically a nucleating site saying, all right, everybody polymerize from here. For actin, there's lots of different places where actin can polymerize because the cell is doing all kinds of different things. So the cell can make these, can assemble either linear structures or branch structures. So it can make bundles, linear actin, or branches. So let's talk about linear unbranched filaments. So a cell is going to regulate where linear unbranched filaments are going to occur, and one of the ways it does that is a protein called formin. So formin acts as a nucleation site. Okay, so actin, if it's above the critical concentration, it'll preferentially start polymerizing here because of the nucleation site. So wherever formin is present, actin can start polymerizing. Because this is very powerful, formin itself is regulated. So formin is regulated by these proteins called Rho proteins. All right, so we'll end there by saying you're going to regulate the places where actin can polymerize, and we're starting with formin before we get to ARP23 and other things. Okay. See you guys Wednesday.
I think because I've been PNing. I've been like part of like.